you very much for the introduction, and let's start. The first question is, can you imagine losing more than 50% of your production? And that can happen if you are not trade, you feel your sugar beet field with when you are affected with this disease. My disease, which I was working on my PhD, was sarcophoral leaf spot. And it's a polycycle disease. That means you have more than one cycle in a season. You can have till 50, it can happen till 50 times. The disease start like a small spot on the leaf, and after that, the spot collapse together, and you can see here the development on the field. At the end, you lose all your photosynthetic part. How that's work? The disease starts with you got the spore come to the leaf. After that, the infection, the fungus grow inside, and after that, you start to see this. What I mentioned, this is more a small spot. And in this spot are the fructification part. This means that they generate the small spore, and the spore start to fly. And so happen many times when the weather or the microclimate is propitious for that. For that, here is my motivation of my roadmap for this dissertation in a single view. To understand the contribution of this work, we must first look at the three fundamental research gaps. First, the digital phytopathometry gap. Current disease assessment is still largely visual and categorical, which is limit the precision, throughput, the reproducibility. In the initial disease signal is inconsistent and downstream the analysis. Whereas the mapping, comparison, or modeling become a fundamental and unreliable. The second one here, the second is the robustness across platform and disease. We know that the disease metric can drift when we change the sensor, we change the platform, we change the resolution or underlying the disease biology. We therefore need methods that remain stable and valid beyond a single sensing configuration and beyond one pathos or one single pathos system. The third one is the sensing the epidemiology integration, like your sensor and epidemiology together. We must remember when we are doing like remote sensing that the pixels is not a process. Forecasting require phase-aware biology grounded target that explicited the link between optical sensing and the actual epidemic dynamic, such as I showed the incubation, the fructification, and the spreading. Addressing these gaps, I have here to address this gap, here are my central research goals: to develop and validation a sensor-based monitoring framework that can detect, quantify, and model phase-specific on this dynamic that I showed you in sugar beet under real field condition. The first goal, I separate that in 5 sequential goals, which each one have a specific intent. The first goal is the cataloging individual plant to establish a stable georeferenced unit that make a longitudinal analysis, allowing to follow the exact same plan over the entire season. The second goal is starting to the disease severity classification. Convert this imaging as a raw imagery into reproducible severity information that align the sensor point of view, enabling consistent quantification at the field scale. The third one is scaling and adaptation across platform to test what is gained or lost when we're moving across sensing configuration from UAV to satellite, changing the resolution, going to close range to the satellite, and checking how are these metrics, if they remain comparable. That the third goal is the transferability to other patho system. To stress, that is my stress test, to test the robustness of the framework on contrasting with other disease. In our case, we work with rhizotonia, Crohn, and rot root, where symptoms are indirect because are in the root, not in the leaf, and the biology is completely different. And the fifth goal, and the most important, was this phase-oriented hybrid modeling, to integrate optical indicator with environmental and inoculum dynamics to forecast the phase-specific behavior in biology and in an interpretability way. Now, we will go going moving forward from the individual plant to the hybrid modeling. I want to show summary or data collection. We have a different integration measurement system that link digital ground truth with multi-platform sensing and environmental drivers. From left to right, I show here, we established a method to collect ground truth. We modernized the field scoring, the normal field scoring, using an RTK tablet, and to get a georeferenced and really accurate information from each plant. We have UAV, of course, mounted with RGB and multispectral cameras and after that we use satellites. We have a multi-sensor platform mounted in a robot, and for the most important, for the forecasting and so on, a microclimate sensor, sport trap, which quantify qPCR. This provide the phase-relevant information that connect observation to epidemic process. Now, I will start with my first model, how plant level cataloging created a stable unit that make all downstream analysis possible. The work is really based with a friend from me, also a PhenoRob PhD student, Maurice Gunda. He was the first author of this publication. I was the co-author. And the motivation here, as I say, is simple. For a longitudinal epidemiology, pixels are not a stable unit. They change with illumination, point of view. So we need better foundation. That means detect individual plant, have it with a really nice georeferencing across all the time, all the vegetation period. So with that, we managed to do that. This slide show how the plant catalog is built from UAV to orthomosaic, from UAV orthomosaic. At the beginning, we have the drone image. We make some kind of segmentation. We apply some peak finding, and after that, we assign a plant ID to each plant, and we can see here from March to October all plant on the field and following with the catalog through the vegetation period. So we got for this goal, we established an individual plant tracking across the full season. There was some small boundary here. The system file under very high, fully canopy cluster when plants are not longer visually separable from above. The second goal, the automatic severity scoring. This work was also published in the Sugar Industry Journal. And the problem here is straightforward. Manual disease scoring is slow, complicated, subjective sometimes, and difficult to scale. So we use at the beginning a CNN to turn a high resolution imagery to a reproducibility severity classes. How we do that? At the beginning, we have to collect our ground truth. So these ground truths need to be strong. So the experimental design was built and specifically for learning from UAV data. in 2020, that is the field from 2020, we flew two, uh, two different fields. You can see here from the left one, and the second was this one, here in the near of Göttingen. Field 1 was a variety trial with inoculation plot. This created a clear disease gradient under the high pressure. And the field 2, we use a spot inoculation. We put we inoculate with the disease in the middle of each plot to produce some kind of heterogeneity so to get in some kind of spatial pattern on the image. I'm gonna show that. We use one for training, and the second one we use only for tests. For scoring, we adapt. We use the KWS scale for this disease, which was from 1 to 9. One was the very healthy plant, and 9 was completely disease, and the number 2 was the first symptom. The KWS scale was developed in the 70s, but was not adapted for use to drone, and they don't show all intermediate class. So we decide to fix that, find in the image from the Nile point of view, image which match this description, and we use that. We base on that for classify our fields. So was on the left side, we can see how we do that. We implemented a very easy LeNet style. We use 2 convolutional activation pooling blocks. It's very easy, followed by a fully connected layer, and after that, a softmax classification to get these 9 classes. We choose this architecture as a robust baseline to ensure stability and comparability across studies. And so we can see here how we can classify. The classifier that here is one of the metric of the result, a confusion matrix. So we can see the confusion show the strong diagonal agreement between the classes, but still existing some uncertainties, reflect the change of forcing gradual symptom because they are kind of ordinal into decreased severity level. And but at the end, we got the first plant scoring for this disease on Sugar Beet. Now I want to show how, as I showed the field we spot inoculating, we can see here, after we run our model, this plant level prediction, how that can be translated into epidemic maps across the field and over the time. We can see here one is the map plant level prediction back into the field geometry. We obtain all the spatial epidemiology. Here you can see the epidemic progression across the season. Initial foci appear here. We can see appear here. That start okay. This is start here where we inoculated. And at the end, we can see how they expand outward, and the final, we can be the broader spatial dispersion according to plots. And this is the first major transformation in my thesis. From raw sensing become a structural spatial temporal disease signal, not just image, but quantified dynamic in the space and time. But now the question is, when we achieve that from plant, we got that first, the level severity classification. The CNN is positioning as a decision support. It's working. It's help for augmenting expert judgment, not replacing them. That is very important for me, that we show that when we have a stable homogeneous severity scoring, we can help the expert on the field to be more homogeneous in their scoring. But for the next goal, can this pipeline survive the change of sensing platform? we can see that. The question is straightforward. When we change the sensing platform and the spatial resolution, does our disease monitoring pipeline still working? This goal is addressed through two publications. One was in a peer-reviewed conference paper, and the other was in a journal article. We explored two different directions. Using translating that to satellite scale, and other one we used to our ground basis robot platform. In both cases, the core question is the same. And in the left side, we can see here or satellite point of view. The satellite, the problem with the satellite, they have different spatial resolution, but also the resolution, the resolution is not different, but the band or the band resolution is different. For that, we're thinking a workaround, because we try to convert our drone data in satellite image first. When we got this, we transform our satellite or drone data into satellite image. We use this artificial image, generated artificial image, for train a model which can be used in satellite image. And we can see that work better. We can see here in our confusion matrix, it's work better. The diagonal classification that they say like is still there, but we have some higher uncertainty here, but that is expected because the satellite, because of the spatial resolution of the satellite, we can sometimes we get noise on the values from the pixel values. Or the satellite which we use was 5 meter, and that is what is one of our idea why it's not so good. The second paper was about using a multi-sensor platform. I'm gonna go faster because here was more the configuration of the platform and using on the field. And we have a multi-sensor platform with different sensors. We have LiDAR, Oster scanner, we have RGB and thermal, and we conduct a field experiment here. The confusion matrix I'm not showing now because, but there was also similar like from the old CNN. But the thing is here is that we got to get that to preserve the structural fidelity here. We tried to use the extent of our sensors, and the adaptation was possible, of course, with some changes. But there are two boundaries here. The work that we use was with only synthetic data. We don't use real satellite data. And about the robot and the UGV and the satellite are a single time point demonstration. They are not. We don't make a multi-temporal tracking between the platform. But let's go for our fourth goal. To provide our pipeline is a robust framework and not a model trained for one disease. We tested the transferability on Rhizotonia crown and rot root. As I mentioned at the beginning, it's a soil-borne systemic disease. The canopy symptoms are indirectly and heterogeneous. So we have a kind of wilting, chlorosis, a collapse of the leaf part, and can be confused with other problems, the drought, also dry drought, and I don't know, nutrient stress. They made that very ideal for a stress test. What we do that, we need to adapt the required rethinking the scoring logic because it's not the same. The normal scoring that is did here in Germany for scoring this disease is at plot level because Rhizotonia created heterogeneous pattern. We can see here the healthy areas adjacent to severity affected areas. And what we do is we need to transform to the plant level, as I did before. At the plant level, we adopt this scoring from original 1 to 9, because it was not possible to have 9 classes here. And we take less classes to try to score better. We take the healthy classes, first symptom classes, wilted classes, and mummified classes. Is when the plant got complete brown, black, and disease. That the pipeline, but the pipeline logic remains the same: plant cataloging, image extraction, classification. But the disease biology is different, and the class structure are really different. It's not moving here. Oh, yeah, here. Once the plant level prediction are the aggregate, the plot level we can compare automatic scoring with the expert assessment. Yeah, here we can see here is we have our input from our model. We did the orthomosaic, the plant cataloging, and the shape file of the plot. And here is the classified plant. We know which level is each plant, and we can see our new model give to us the plot level scoring. We have here 2 confusion matrices. One is from the classic plant-based. We can see here also the diagonal, but the plot basis is what we got very good result in the test data set. And so what we do here, we maximize the different stress tests. We show that the more our idea is work in other disease. And it's possible also to capture information from below ground. As I say, we get the same pipeline but different biology. And now we are now reach the final goal of my thesis, the hybrid epidemiology modeling. This work has been accepted for publication in Phytopathology. It's already published, the work. This is where we connect everything: plant catalog, severity signal, and environmental and spore data into a phase-specific forecasting framework. How that work? For that data stream, we have different kind of data. We have the drone data, the multispectral data. We have the plant catalog and also the disease scoring. We have climate data. And here, what we add here is we use other mechanistic model, which were weather-based mechanistic model, also as input for our models. And we got also export the default dynamic. And the core of the approach, instead of one model for entire epidemic, we structured the analysis around 4 biological phases. We can see each phase has its own target variable and its own domain driver. Incubation is driven by the cumulative evapotranspiration, and we have different variables here. What we do is first feature selection, and we train models, and we test different kind of hybridation here. Hybridation at the future levels, hybridation after residual, hybridation combine prediction from mechanistic model, prediction from our data-driven models, and so on. For the data collection, we conducted field experiment for 3-year field experiment. We have spore traps in each plot. We have micro weather sensors, and we have also extra experiment where we try to understand the disease dissemination, where we inoculated on the middle, and we have spore traps in each corner of the field, of each plot, so we can see in which direction the spore are flying. So with that we can go the information of the dissemination from the incubation period on all of that. So after that, we did make a data construction. We used the plant cataloging for extract information from each plant of the field, and we put all together like a 3D cube. So after that, we train models, we test the different hybridation, and we can see here that the hybridation reduce for us the error. The error are a reduction. We have an average of 21.6 reduction of error comparing a very basic model with the data and a hybridated model. In the right side, we show here is which for each epidemiological phase, which feature is important. We can see here that for the incubation or for the disease severity, the evapotranspiration have some very high importance. For the amount of spores, some mechanistic model have a very high importance, but still the NDVI data still having some importance. And how that at the end we have what we got here. We implement the first UAV climate spore mechanistic integration for this disease. We did it at a phase specific with a phase specific architecture. We test three different kind of hybridation, and we got up to 39 reduction of error. And to close, I summarize here the dissertation in from these 5 goals, each with a clear contribution. Also, I showed the boundaries because always have boundaries and what we can do in the future. First of all, about the cataloging, we established a plant ID for a longitudinal tracking, moving from pixel to established biological unit. The boundary here is canopy cluster. Where the limitation become really, really hard. When the leaf touch together, then it's a problem to separate that. The next step is try maybe some temporal instance segmentation for dense canopy also on. For the severity, we deliver the reproducible severity classification aligning to the UAV top-down sensor view. We got a digital scoring. The boundaries here is the transition between symptoms, where the label are really having some ambiguous on some kind of problem between two the scale. The next step is trying to transform this from continuous severity, trying to get that in a continuous severity regression, trying to understand what happened in the middle. Also, some kind of active learning labeling could be helping here. And the third scale, we address a multi-scale sensing into 2 directions. We do the UNet the conversion from satellite. We test a multi-sensor platform. And the boundary here are limited. The matches the satellite data validation and the sensitive of fusion to. I didn't mention that, but also the co-registration of different sensors for the multi-sensor platform was very complicated, but they are better way to do that. And the next step is, as I mentioned, that is match the satellite acquisition and a stronger cross-site calibration. About the transfer, we stress the tested robustness on Rhizotonia, showing the framework can adapt to contrasting the disease biology. The boundaries is clear. One pathosystem is not universal and it's not universal proof. The next step is transfer learning across region and additional having some more than one disease, because I show only Cercospora, only Rhizotonia, but I don't got all the disease or the both disease together. And about the hybrid modeling, we link the sensing with epidemiological phases and achieving error reduction. The boundary was here is this reminds knowledge that's the hybridation that which I test here was a very basic kind of hybridation. This knowledge guided machine learning was not a fully mechanistic or causal. The next step so is the testing in different kind of hybridation or more robust hybridation, and also maybe in the future is trying to get hybrid digital twins of that. So overall, the thesis drives a modular path from plant unit to reproducible quantification to multi-platform robustness, and finally to a phase-aware forecasting. So thank you very much, and I am happy if I got some questions.